were talking about DNA viruses. Today we'll begin with RNA virus. Okay. So the family of viruses that we're going to study today are PICORNAVIRUSES. As the name suggests, Pico means small and there's an RNA there. You really have to look for RNA or DNA, you'll find it there. And viruses, of course. This is one of the largest families of viruses, and it does include some of important viruses of human or animal disease. And remember, most of these viruses are naked. What does that mean? Yeah. They're not encapsulated. There's no envelope there. There's no envelope there. And what is the difference? What is the difference between a virus that is naked as compared to a virus that's say it again? Virulence. Which one is more virulent? It's more virulent, like? Yeah. What?
Just how easily it can be transmitted. Ones with capsules require to stay in water, while ones that don't have capsules.
Easily transmitted. That you're saying the one naked are easily transmitted?
Yeah.
Okay, yeah.
The ones capsulated, they have more of a immuno effect because they're not approaching on the capsule.
Okay, so maybe there are some kind of glycoproteins or markers on envelope, so they may provoke a good immune response. Yes.
Would they be hard to treat since they're encapsulated?
Harder to treat. Okay. Like, which virus are you talking about? Which virus is harder to treat? Which virus do you have in mind? You say it's harder to treat. Or just guessing? Okay. All right. Well, let me bring one important pathogenic mechanism for these viruses. Remember that normally, if you remember replication of viruses that we did do in the journal microbiology, remember I said the other day as well, you need to go back and read that chapter as well. Journal microbiology, we talked about viruses picking up a layer. A typical virus that buds off is the one that picks up the outer cell membrane of the cell. That's why you see this typical kind of envelope that's picked up by the viruses. And these viruses are those that will bud off and remain persistent. Most of these viruses will call pathogenicity in terms of persistence as compared to the naked one, because the naked ones are living. They will just lyse the cell. So once you lyse the cell, you cannot persist in the cell. Right? So this is one of the mechanisms where the naked viruses have this capability to lyse the cell. So most of these infections are lytic, not in terms of persistent or latent. So these are important mechanisms that you always have to keep in mind for these special viruses. And again, as I said the other day, virus is nothing but a bad news wrapped up in a protein. So in this case, the bad news is RNA. So RNA is there, you can see an RNA virus. And this is actually a list of different viruses that are included in PICORNAVIRIDAE family. And actually, I'm not going to cover, I actually also deleted some of the viruses that I think that are not of our concern for this class, but your book has mentioned quite a few viruses. Enterovirus, the most important virus in this group is an enterovirus, and then a rhinovirus. And rhinovirus is the one that gives you common cold. Common cold. Common cold is a rhinovirus. And then again, we have another virus. I'm going to take hepatitis tomorrow, very important virus. And I've already asked Dr. Mukherjee to give me his class time. So immediately after the test, we're going to begin. This is going to be 2 hours at least, because we're going to cover a big family of hepatitis viruses. It's very important and very clinically related to you as well. You can see in enterovirus, again, there are different kind of viruses. Polioviruses have 3 strains that we normally vaccinate. Then there is a COXACKIECIRUS, and Coxsackie is like a small town in upstate New York. And that's where it was originally found out, and it was named after that town, COXSACKIECIRUS. There are 2 formats, A and B. Have you heard of any disease caused by COXSACKIECIRUS? No? Have you heard of hand, foot, and mouth disease? Very common in children. It is a differential diagnosis for chickenpox. Many a time mothers notice it. It's a typical one caused by Coxsackie, and there are quite a few. I have a couple of other things to discuss in this virus. I just have one slide for this virus. Echovirus, again important. Enterovirus, of course, is a bigger group, but we can be a little bit specific. And then rhinoviruses, and then finally hepatitis A virus. Remember, the hepatitis virus can be a DNA virus as well, like hepatitis B. So we'll take that as a group tomorrow. I'll just want to tell you that all these viruses have something in common. And the most important thing that you have to keep in mind that typically, as I said the other day, the viruses will evoke a cell-mediated response. But in this case, those cell-mediated response may be evoked, but this is humoral immunity that's more important to take care of these viruses. So that's one major thing that you have to remember, the significance of humoral immunity in these viruses, especially enteroviruses. And again, because hepatitis A virus also takes an enteral route as compared to IV route that hepatitis B or other kind of routes that are available. For hepatitis A is infectious, so this most of the time the hepatitis virus enters into your system with the food. Okay, let's talk about a bit of special properties of these viruses. So you can see from here, they are small, and they have a single-stranded positive RNA genome, so they are ready to go. They're just going to go and start multiplying because they have a positive RNA genome. And another important thing you have to keep in mind that many a time these viruses have picked up a tropism because of their ability to survive in that particular environment. For example, if we label a virus as enterovirus, and this virus has to enter in your intestine, obviously it's going to survive all those conditions there in the intestinal tract. So you can see from here, they are pretty much resistant to pH 3 to 9, so they have wide spectrum to work because they can survive in the stomach, they're going to survive in duodenum as well. They can also resist detergents, sewage treatment, and heat, so they would not be killed because many a time it's just like an ice cream. So you really want to keep an ice cream in the freezer. If you put it in the fridge, it's going to melt down. So if a virus has an envelope, so it's going to melt down or break apart if you don't give the right temperature. So in this case, since they are naked, so they can pretty much stand everything, every kind of adverse conditions that may come in the tract. By the same token, rhinovirus, as the word suggests, is more like an upper respiratory tract kind of an infection that is labile at acidic pH. So this means if you ingest this virus, it would not cause any disease because it has to go into your respiratory tract to cause disease. Otherwise, it's going to be destroyed. The optimal growth again is 33 degrees C. Remember, many a time these viruses will go for upper respiratory tract because the upper respiratory tract has a little bit less temperature as compared to the lower respiratory tract because lower respiratory tract will have the same temperature as their body temperature. But since there's a lot of ventilation taking place, so they want to sit near your upper respiratory tract, pharynx, nose, and so on and so forth. Right? Does it make sense? So we have this 33 degree. Genome is a messenger RNA, and naked genome is obviously sufficient for infection. Virus replicates in cytoplasm, and viral RNA is translated into polyprotein, which is then cleaved. So it's typical kind of replication of virus that happens with these. And as I said earlier as well, since they are to lyse the cell before they bud off and cause infection to other, so these viruses are cytolytic. They are going to lyse the cell, which is going to provide them with all this machinery to divide. As compared to the one with envelope, they can easily bud off or remain in the cell and persist or become latent later on. Okay, now let me show. This is like a detail of whatever is here in this figure. I'm going to concentrate on this figure. And many times I realize that there's too much information on these kind of presentation. And since I use this as a handout as well, and use this for posting on your live text, so many a time you'll find more information than actually should be presented in a typical PowerPoint presentation in the lecture. But anyway, let's concentrate on this one again. Pathogenesis of a typical enterovirus infection as a whole. And remember, these are the basic things you have to keep in mind. It will be easier for you, because when we say pathogenesis, we're talking about the entry of virus, number one. Entry of virus, and where does it go? And then the typical clinical picture will be dependent upon which organ of the body does this virus invade. So if you have a respiratory tract, you have symptoms of upper respiratory tract. If it's a gastrointestinal tract, you have symptoms of gastrointestinal tract. So symptoms of disease are easy for us to understand once we know where is this organism going to end. If it's going to end in the liver, obviously we will have problems with the liver, right? And then it will cause tissue injury and obviously make those organs dysfunctional. So you can then come back and look at the altered function of that particular organ. For example, if there's one virus and that infects liver, it's going to cause lysis of the liver cell. So all the normal functions of liver are going to be jeopardized, and we are going to find those relevant tests to find out what are the functions of liver and how do we really assess that. Now, as you can see from here, these viruses are going to go for enteral route, and enteral route means oral. So these viruses have tropism, are liking to enter, and remember these viruses are usually very keen to go for mucosa, because skin is pretty much resistant, so they're going to go for mucosa, because many times on mucosal cells we have corresponding receptors, and they will find a nice place to really move into the system. So rhino, echo, Coxsackie, polio, hepatitis A virus. HAV stands for hepatitis A virus, and HBV obviously will be hepatitis B virus. We'll come to that tomorrow. And you can see that they will come and replicate in upper respiratory tract. So they will go and attach in oropharynx most of the time. So you'll have mild discomfort of the throat, sore throat, or a little bit of inflammation that may happen in upper respiratory tract. That's a typical of enteroviruses. But once they go into the system, they are going to spread out and invade and enter circulation. So immediately, these viruses will go into our blood. This also means that many times we are all exposed to these viruses. So you or me may already be immunized by nature. That we already got these viruses hanging around here and there depending upon their dose or immunity, and we end up recognizing them and we end up building up viremia. So once we have a viremia, something circulating in the blood, so obviously by our typical definition of humoral immunity, antibody cells making antibodies are going to see it and produce antibodies. So they have a chance of being recognized or being targeted by antibodies because they are in fluids, as compared to those viruses which will enter into cells. For example, we talked about those STDs, and they actually were the one which actually find the place in mucosal cells and difficult for the other immune responses to come over. So the primary response was cell-mediated immunity. And in this case, you can see typical primary viremia, but antibodies can block that. And that depends upon your prior exposure to these viruses of the strain, or you've been vaccinated. So your vaccination status, your immunity, and your being, because many a time, remember, we are all silently infected with these viruses. We don't show any disease, we are asymptomatic, and we don't know it, but we may have been exposed to these viruses, and we may have titers of antibodies against those specific viruses. So what we are talking about over here is that if such is the case, then the antibodies can block that. And this blockage will be, these antibodies, you should remember one of the functions of the antibodies will be to block the entry of these viruses into the target tissue. So don't let secondary viremia happen. Because primary viremia is like they will pick up these virus immediately by the first kind of circulation that will happen. And if there is no antibody there, so they will have a chance to enter anywhere. And then these viruses may have affinities for other organs as well. They went and replicated in the lymph nodes over here or structure because they didn't get a chance to go into the blood. Once they are in the blood, then the blood can take them wherever they really like to go. So the most important organs that are exposed, of course, are going to be skin. That's why many times you see skin lesions. So it's the other way around. Sometimes remember that you see these primary lesions at the sites of primary inoculum, like those STDs. In this case, it's the other way around. So you see it goes into blood, and the blood takes it to generalize. So these kind of lesions will be generalized. You'll see a typical like smallpox. Now, skin, again, muscle, brain, meninges, and liver. These are some of the commonest target tissue. And if you remember that these are the targets that are picked up by these kind of PICORNAVIRUSES, you can pretty much determine as to what is going to be the clinical condition. Right? For example, if it's in the skin, and that's for ACO and COXACKIE virus, you will see hand, foot, and mouth disease. So you'll see typical kind of these lesions or rash on the skin, because it shows that the viruses are running in your system in the blood. And many a time you see rash and herpangina. Herpangina is kind of a lesion that we used to think is like a herpes kind of a lesion, but it's not herpes. Remember I said that it's easy for me to show you all the slides of CDC where they had already diagnosed the case. But if you were to see it for the first time, things are not that easy because there should be other differential diagnoses. So one of the lesions that look like herpes could be because of another virus, and that's going to be ACO virus or COXACKIE virus causing herpes-like lesions. We call herpangina. Herpangina, that's different from angina pectoris or coronary angina, right? Now, as far as muscle is concerned, many times remember if we have viral infection, we have muscle aches and pain. So muscle aches and pains, well, the viruses have reached over there and they are causing tissue injury. Typical again is ACO and COXACKIE A and B. The most important thing you have to realize is that they can also involve muscle of the heart. They can cause myocarditis or they can cause pericarditis. And sometimes very common, people have acute chest pain and they think that they have heart attack, and it turns out to be an infection with one of these viruses that's called pleurodenia. So the pleural cavities, if you remember those two pleural layers, they get inflamed and they rub against each other and they give you a kind of a picture as you may get in heart attack, for example, or myocardial infarction. And it happens very commonly to people that think that they have a heart attack and it turns out to be a viral infection. So that's why the most difficult differential diagnosis in medicine is differential diagnosis for acute chest pain. It could be anything, right? But of course they don't take the chances. They will treat you as if you are going through a heart attack, because that's the worst that could happen to you. And then they will rule that out and then go for some other benign conditions. So pleurodynia is important. Dynia is with the pain. And in these cases, pleurodynia, what happens is that this virus infects the skeletal muscles. You know, these obliques and transverses, all these kind of muscles that are there between the intercostal muscles. So they infect those intercostal muscles. And that's where the pain is coming from. All right. Then of course, brain, very important for polio. Polio is again an enterovirus. And then again, in polio and even Coxsackie, you may get a paralytic disease. It's been eliminated, but of course, the vaccination started in somewhere in the 1960s. Those people born at that time, even in US, you know, they still have those paralysis of usually one leg or one arm. Very, very common, and it's still going on in rest of the world. And the problem was because WHO is still trying to vaccinate all the people, and with all those difficulties that they may have to reach out to people living in deserts, living in villages, there are still occurrences of polio in different parts of the world. Encephalitis again. Meninges, typically any virus can go and you got meningitis. It doesn't really matter. We usually call this, let me put it this way, we call it aseptic meningitis. So you see the typical features of meningitis, irritation of meninges. You know, you do the head raising test. You just want to let the patient lie over like this and want to raise the head. You will not be able to bend his knee. It's kind of a change. You ask him to touch his skin with a test, he would not be able to do it because there is a strictures over there and there is inflammation. So that will be neck stiffness, typically neck stiffness caused by meningeal irritation. But that can happen because, most likely because of bacteria. But if you do the spinal tap, and you don't see any bacteria, the chances are there because of virus. Remember, we have a challenge of culturing the virus. So viruses can cause meningitis. We call them aseptical, typically because we cannot find bacteria there. Doesn't mean the meningitis is not there, but it is there. And finally, if it reaches liver, like HIV, we get hepatitis. And liver is a major organ, so if your liver is damaged, then we have all those complications in digestion, in making plasma protein. You remember all the functions of liver. All those functions of the liver are going to be jeopardized, right? And details of whatever I just talked about are here. Right. Any questions on this slide before I move to the next one? Are you preparing for test tomorrow, or are you with me? Half and half? Multitasking. All right, good luck. Okay, all right. Now, as far as epidemiology is concerned, again, there are many things that we keep in mind. Most of the time, the nature of disease correlates with the age of the person. Remember, if it's an enterovirus, it's in a baby, and the baby is growing, polio is important. And if it happens later part of his life, then we don't really care much. And we also look at the vaccination status of the patient. And many times, as you can see, the person may be asymptomatic like most of us. Doesn't mean that we are not exposed to all these kind of coronaviruses. We must have been. But we may have picked up active immunity. These are the likely chances, right? Or because we all have been vaccinated. So these are the things that we usually keep in mind. But nevertheless, there are always viral shedding. Why there is viral shedding? Why there is viral shedding in PICORNAVIruses? Yeah. Because they are lytic, right? They have to kill the cell and then shed off. There's no budding. So typical shedding will take place because they're going to break from the cell and disperse out. Right? For example, if they are growing in your upper respiratory tract, so you are continuously shedding off, passing on to other people. And many times, some of these viruses, especially rhinoviruses, common cold, it's going to go in droplet infection, it's going to infect others as well. And that's very common with enteroviruses as well. In some of the situations, like for example, if you happen to work in a daycare center, right, or some kind. You have a question?
So the virus shedding, is that like the point where the person is like highly infectious? Because like there's like the one we found yesterday, there's two stages here, and after a certain period of time, you get a virus, and that's when it's not really infectious.
Well, they're highly infectious anyway. Once the viruses are there in open, for example, virus shedding means, let's see, like you are in the, let's say, daycare center for the babies, and these babies are obviously, if their diapers are not changed, or if there are quite a few babies at the same time, so they may get it from each other because viruses are continuously being shed out in the feces, right? Or if you are swimming and viruses shed out over there, or if you are exposed to a closed area and one person is infected with that, the chances are that you are spreading the virus. So a person doesn't really, remember we talked about those sexy viruses where you really have to have this, transmission through sex. In this case, they are there open for most of the people to get it. The physicians are going to get it from the patients, mothers are going to get it from their babies, those people working in daycare centers are going to get it from those children, schools. So all these, that's why the most important thing you want to consider is the age of the person. So the other challenge is that many times if the baby's their immune system is not that developed, they'll show more of a clinical feature as compared to yourself, for example, because you already have a patent or competent immune system. So virus shedding basically means that virus is there in atmosphere, right? And it's going to be shed out from wherever it's divided. Right? The most important thing virus shedding will be especially in upper respiratory tract infection. For example, if it sheds out from the liver, right? So where is it going to go? Right? It's still going to go in blood, but it's going to be contained within the patient. Unless and until if there is a sharing of needle, or a blood transfusion, or things of that nature. Okay.
So, right?
Sorry?
You did HERS yesterday, right?
Yes.
And it was just that the virus did in the saliva and in the feces and stuff.
Yes.
That person their infections but not to a certain extent that person with it in their lungs and just breathe it out.
Yes. So those viruses that are shed up in aerosol will find a larger area of distribution as compared to those viruses which are going to be shed up by saliva, semen, vaginal, because we really have to come in contact with that. Does that make sense? Anyway, another important thing is this virion is resistant to environmental conditions, so detergents, acid, drying, all these are not going to work because these viruses are pretty stable and they are stable even in your high acid or high alkaline juices in your intestinal tract. So they're pretty much stable there. The other thing is that many a time detergents act because they are lipid in the wall of the envelope. That's another thing. Many times all of these detergents and all of these antiviral things are important if there is a lipid in the envelope. If there is no lipid in the envelope, these detergents are of no use because that's like a genome, right? They are naked. So all these things are not going to do anything. So that's another thing you have to keep in mind. The detergents are not going to work, and drying also, they're going to stay pretty much viable even in dry states. Okay, now as far as the transmission is concerned, how many of you are fan of oysters, shellfish? Well, these are one of the sources of enteroviruses. They say that, typical outbreak that you will see in this country involving many people or oyster or shellfish contamination. And this is very common, especially for enteroviruses, right? But you can see from here, many times, regardless of whatever is going on, because remember, water, we are exposed to this water. You will think, well, I just take this water from, a bottled water, and the different types of treatment they do with the water. For example, in a typical case they give that if you want to play golf, and they obviously are going to irrigate all those fields over there, so they are going to irrigate those fields with treated water. Do you think they're going to irrigate all those fields then with the treated water? No. So once you go early morning, so you are exposed to all those viruses which are there, right? Or swimming. Right? So you actually come in contact knowingly or unknowingly, where basically you really get your share of viruses coming to you. Or sometimes if the sewage treatment, because many a time, whatever goes in the sewage is again recycled. So water, you're getting water from the sewage. They're going to treat it obviously, but it's a recycle, especially in bigger cities. I don't know about Chicago, but I know there was a report many years ago that of all the states, the highest radioactivity, the highest amount of radioactivity is present in Chicago water. So you can go on the water side and see radioactivity levels permissible. And there are obviously many reasons for that. That's why many people want to go and move to Montana, or I know that there is a pharmacy school there though. So there are issues there that the CDC or public health really have to take care how to dispose human fecal matter. And before actually it goes to the sewage, for example, all those nurses and all those people are handling, changing diapers of the babies. So it's rich with enteroviruses. So they usually get exposed to that. So the thing is that all these things, the typical orophycal route, and from one child to another, from one baby to another, is very common. So water supply, hand, all these things basically add on to your share of enterovirus, and this is how the enteroviruses are typically transmitted. Right? So we cannot really sit idle and say, well, I'm not going to get exposed to enteroviruses. We do, and we are. The good thing is that we may have picked up a good immune response. Okay? Okay, this is how an electron micrograph of a polio virus, so you can see. Imagine those people who saw it in the first time, they weren't even thinking that they are microorganisms. They look like particle crystals, very symmetrical, and they have a defined icosahedral shape. If you see, I think they're like a diamond shape with many sides and many, kind of symmetry. So typical of polio virus, you can see from here. And of course, there are many strains for that. We usually talk of 1, 2, 3 strains. These are the commonest strains that we usually get vaccinated against. Okay, now I already talked about that. Fecal-oral route, poor hygiene, dirty diapers, daycare centers. You can ingest it, food and water. And then again, we talked about shellfish and oyster. There have been reports that they do show a very high number of enteroviruses. And remember the heating, if it is half cooked or uncooked, I don't know how do the people eat it, but of course you are going to get a lot of enteroviruses from there. And for rhinoviruses, again, inhalation of infections, aerosols that are going to go. And remember, the aerosol spreads is usually for those viruses, those which are very small. These, you're talking about 20 to 30 nanometers. Remember the size of the very first lecture we had in physiology was talking about the sizes of cells and sizes of bacteria, viruses. So you should have pretty much figure in your mind as to what these sizes mean. Micrometer, nanometer, right? And if you remember the other day I told you about, for example, mycoplasma. Right? I did mention about the size of mycoplasma. How many of you remember that? What is the size of mycoplasma? And there was a very important thing that we discussed. Yeah, right. Micrograms, you said? Micrometers. Okay. So remember, the challenge was, since they can pass through that 0.45 micrometers, the microbiologists thought that they are viruses. So they were so small, because that's the filter we use to filter viruses. So we filter one of the like sterilization or disinfection that we normally do is pass this microorganism through a pore size. So if you know, like, for example, the size of, what is the size of RBC?
7 microns.
Say again?
7 microns.
Yeah, about 5 to 7 micrometers. So what about neutrophils? Obviously bigger, it's about like 10 to 12. Remember, these sizes are important because once you say the neutrophils are going to phagocytose those bacteria, so they are going to phagocytose smaller things, right? So bacteria cannot be larger than the neutrophils. Does it make sense? So anyway, in this case, the infectious aerosols are very small, 10 to 20 nanometers of size. That again will also allow them to easily pass in our system and appear in blood. That's another thing. Because remember, we talked about tight junctions. And those tight junctions, zona occludens and others had these kind of gap junctions where things open and close. And if the size is very small, it can easily pass through. So viruses have a better chance of passing through those intercellular junctions because of their size. All right. Okay, then who is at risk? Again, we did talk about that. We are worried about younger children because many a time, well, the days of polio are gone, especially for this country, because for the last 35, 37 years, people have been vaccinated. And you may see some of the older people, I don't know whether you remember, when we were medical students, we used to be taught from one of the book of physiology, and it was called Guyton's Physiology. And this Arthur Guyton was a professor in University of San Francisco, and he was suffering from polio. So it was like people born before 1962, before actually polio vaccination actually was approved by FDA. Anyway, the other children may be at risk, and because they show newborns and neonates are highest risk for serious COXSACKIECIRUSES. And what does COXSACKIECIRUS cause? I just told you. What is the commonest clinical condition that we come across? Foot and mouth disease, very common for this virus, and Coxsackie especially. Okay, now they do have a worldwide distribution, you can see, and WHO do claim that they have eradicated for most part of the world. But remember, since we are living in an age where people sitting in one part of Africa can fly to US and they're going to be here and bring everything from there. Right? So this is like easier for transmission of any virus from one place, no matter how remote, to this continent. So still they are trying to make sure that they do vaccinate each and every child. And the challenges usually are that it's difficult to reach out, and then the most important logistic challenges, you have to make sure that you keep those vaccines frozen, because if you break that link, the cold chain link, then the vaccine is going to lose its efficacy. So that's another challenge, to really deliver those vaccines at those remote places where there will not be any refrigerated vehicles and so on and so forth. These diseases are more common in summer, most of the enteroviruses. And for modes of control, we have two choices. We have a live oral polio vaccine, and then we have an inactivated polio vaccine. What is the difference between, obviously, a live and inactivated trivalent polio vaccine? Interestingly, one was developed by an American scientist and other was developed by a Soviet scientist. So there was always a kind of a fight between these two. Actually, this is Sabin and Salk. These are the two people who developed that. But what is the difference between live and inactivated polio vaccine? This is something that we already covered in general microbiology. It's going to be very significant to have an impact on the immune system. Yes. What does that mean?
Okay, it has an antigenic purpose because it's not viral.
Okay, well, it's an easier thing that you can say. Yes.
The live one will cause active immunity, where you have to take your own antibodies, whereas the inactivated one will cause you may have an antibody present.
All right. Remember, one is, this is live and other is dead. As simple as that. So you're giving a person a live polio, then of course there's going to be arguments. What if you are suffering from HIV? So if you're suffering from HIV and you are given a live polio, so the chances are you may get the disease. So that's going to test your immune system. So you have to be very careful. So that's a little bit less safer as compared to the other one. The other one is dead. It's not, say, inactivated or attenuated. This means that they would merely kill it. And it's such a mutant that it would never ever cause infection, as compared to the former one, which may cause infection. And fourth, they are both trivalent because there are 3 important strains of polio 1, 2, 3, and they're all being designed towards that. And this actually is one of the success stories, they say, of immunology, of the medicine, that if there was any good vaccine made ever, this was it, polio vaccine. And then, of course, 40 years ago or 37 years ago, but these two scientists, Sabin over here is Sa, and basically at that time the challenge was, of course, to combat poliomyelitis. And Sa was the first who tested in 1952, and then later on in 1955 we came up with an inactivated dead one. And of course, it was approved in 1962, and most of the time, and you are right, basically, remember active immunity. The other important aspect of that is that, remember I said that for enteroviruses, humoral immunity is more important than cell-mediated immunity. Right? If you are given a, let's say, a live virus, of course you're going to have an active immunity. The challenge will be, for example, that you develop those antibodies and then you may need to have booster doses because your antibody titers can go down, right? As compared to the other one where you already are giving a passive immunity, right? And the chances are that you may need just one shot. So this is one of the logistical difference that we have, but of course there are pros and cons for both of these groups. And he was a Soviet scientist, but he actually came and did his MD from New York. I don't know when he did develop his vaccine. Anyway, I don't want to go in the clinical stage of poliovirus, but you can read it at your own, because this is more like a clinical condition. The chances are, as I said earlier, you would never ever see that. And but just remember that it could be 4 kind of outcomes. You can be asymptomatic illness, you can have a minor illness, you can have a non-paralytic illness, or you have a major illness. But you can see the chances are very remote, especially after vaccination. The only other thing that people think is that those people, especially who have passed 50 or 60, and their cells, by aging process are degenerating, and they cannot really control, their immunity is also degenerating, they may get a chance to be exposed to these enterovirus, and they may show some kind of neurological disabilities. Okay, typical in this case, if you remember the other day we discussed about herpes as well, they tend to move from blood to anterior horn cells, and depending upon which part of the nervous system they go and invade. If they invade, let's say, the anterior horn cell of one side of body, for example, is left leg, and all those nerves supplying to those parts of the muscle will be paralyzed. If you get a bulbar like medulla oblongata infection, so you may have paralysis for the whole body. That actually depends. The most important thing that we need to keep in mind is that we don't want this virus to go and invade the central nervous system, because if it does invade central nervous system, we may have problems. But if it goes for anterior horn cells applying to one leg or one arm, that's a typical scenario that we normally see. All right. Now, I think two more slides. One for Coxsackie virus we already discussed. Group A can cause herpangina. We talked about herpangina is a herpes type lesion, differential diagnosis of herpes simplex or hand, foot, mouth disease, and can also cause pharyngitis because it wants to replicate in upper respiratory tract. Group B can cause myocarditis and fluoridynia. Remember, it's like a differential diagnosis of acute chest pain. A person may be suffering from Coxsackie virus, and that causes infection of intercostal muscle, and you got this like stabbing pain. A person feels that he's going through a heart attack, for example, and that turns out to be a Coxsackie B virus infection. Typical infection is by nasopharyngeal mucosa, because you got these aerosols there. The incidence is very high during summer and fall. And finally, rhinoviruses, common cause of common cold. Just want to make sure that you understand the difference between common cold and influenza. Right, so that's the difference, because many times we think flu and common cold are the same thing. Well, laymen may say that, but we're going to study them differently. Common cold is like typically by rhinoviruses, right? And rhinoviruses are from the enteroviruses group, or from the PICORNAVIRIDAE group, right? So these are the typical viruses that cause common cold. They are not that bad as compared to influenza. And the problem you can see from here, they may have hundreds of serotypes. So there's no point getting vaccinated for common cold because there will be too many vaccines available. The most important thing that you have to realize and understand in pathogenesis of common cold is that for 80% of these viruses, they have a receptor, and this is an ICAM1 receptor. What is an ICAM1 receptor? We did discuss that in immunology. What is an ICAM? Yeah. It's an adhesion molecule. Well, remember the same old, many times I ask the question, the answer is already there. Almost like 100% of the time, this receptor has been identified as a member of immunoglobulin superfamily, which is expressed by epithelia, fibroblasts, and B lymphocyte cells. As simple as that. The definition is already there. So these are adhesion molecules, intercellular adhesion molecules. So this is something that will give an edge to these viruses, and these are also used by the Coxsackie viruses. So basically, we can easily go for epithelia, fibroblasts, and B lymphoblast type of cells because they share the homology for that virus. And again, pretty much the same. They are unable to replicate in gastrointestinal tract because they cannot stand those temperatures, they cannot stand those acids and those alkaline juices. And interestingly, some of the research says that infection can be initiated by as little as one infectious viral particle. Well, I don't know about that, but that's what the research shows, that you can get it. So it can increase your chances. And if you can get infected by one particle, and normally if a person, for example, sneezes, he's going to release 500 to 1,000 infectious particles. So you can see the probability, the chances of you getting the infection from these viruses. Again, virus shedding is important, and many a times the question is, why do we show that kind of running nose or a typical a clinical condition where we have to take antihistaminics? The reason is because the infected cells release bradykinin and histamine. So the cells infected by these viruses, they release. And remember, these are normally present over there, right? And these are vasoconstrictors or vasodilators. These bradykinin and histamine are vasoconstrictor or vasodilators? Vasodilators, right? Because that's why you take antihistaminic to constrict them. As simple as that. Okay. But it took you a while to answer that. All right. Okay. As far as pathogenesis is concerned, immunity to these is transient because there are many serotypes, so there's nothing you can do about that. And finally, cell-mediated immunity is not likely to play an important role in controlling rhinoviruses. That's why the role of humoral antibodies. Because if there are many types of antigen being exposed, so our bodies are not going to waste energy of making all those antibodies for no use. So the body's really clever in that. It's going to waste all those proteins there. And typical clinical syndrome, everybody goes through that. We pretty much know what happens in common cold and what are the lab diagnosis is not necessary. No need for you to be diagnosed for a common cold, and the treatment, of course, is available for all those over-the-counter remedies which can work. If you want to take antiviral therapy, they are not effective to begin with, and number two, they are all very expensive. Okay? The important thing, I just want to say one last thing before I finish, is that sometimes people advise the use of interferons. The problem with interferon is that many times interferon is going to activate your whole immune system, and you may have a clinical condition because of the side effect of interferon. So interferon is not going to help you, but of course it can be marketed, it is marketed, and sometimes people do use it. Any questions? Okay, so we'll have test tomorrow, sharp 8 a.m. And remember, there are 75 questions, so I'm going to bring starting time, finishing time. So 75 questions will be done in 110 minutes. So, and I already announced 15 lectures. I'll pick up 5 questions from each lecture. Okay? There's some announcements.
Before you guys run off, anybody that's handed in a doctor's note any time in the past couple of weeks, either to miss an exam or to miss a class, you need to go to student activities or student